I'll moderate the discussion at the end of the talk, but for those of you that are online, please post your questions in the chat ahead of time. If we have enough time, you can also raise your hand at the end of the talk. It is my pleasure to introduce Dr. Phil de Jager, who is a Weil-Granat Professor of Neurology at Columbia University. He directs the Columbia Multiple Sclerosis Center. He's also the chief of Neuroimmunology, and he directs the Center for Translational and Computational Neuroimmunology. He's also the deputy director of the Taub Institute for research on Alzheimer's disease and aging brain. Phil applies modern methods of neuroimmunology, statistical genetics, and systems biology to enhance our understanding of complex immune-mediated neurological disorders and degenerative diseases. He led a number of very large genetic studies, including a recent study of over 100,000 individuals comprehensively mapping susceptibility to multiple sclerosis and demonstrating that different immune cells contribute to the disease process beyond just T cells, and identified also a number of potential drug targets in that study. He's also a leader of the NIA-funded Alzheimer's Disease Functional Genomics Consortium, and he's leading the efforts at the construction of this flagship map, functional genomic map of aging brain in humans. So we're very much pleased to have him here to join our seminars, and we're very much looking forward to your talk, Phil.
Thank you, Professor, for the kind introduction. So, without further ado, we'll get started. These are my disclosures. None of them are pertinent to this talk. So I'll be sharing with you today three different vignettes. First, on multiple sclerosis, what we've done so far to go from the gene discovery efforts that Christophe mentioned towards primary prevention, which is one, of course, of the most important roles we can have. I'll talk a little bit about functional genomics and Alzheimer's disease in the second vignette. And then the third one, introduce some newer work that is where we're trying to map trajectories of immunosenescence in diverse populations to try to get a better handle on that source of variability in the aging individual. So the MSGWAS that we did now several years ago was the largest at the time and still remains the biggest so far, although we're now beginning to do a larger one. At the time, we did basically a large discovery study and then did a deep replication in another 20,000 subjects for a total of over 47,000 cases and 68,000 controls. So with this power, for this particular trait, we were well powered and we identified over 235 genetic variants. So they're shown here. Obviously, we're not going to go into each of them separately. The circus diagram. The circus diagram here sort of shows all the different loci that we mapped with susceptibility to MS. And again, this is published a couple of years ago, so you can take a look there. There was a parallel effort looking at the less common variants with the exome chip that was published by our group as well a year later. But the major features here is that the genetic architecture of the disease is primarily driven by common variants of modest effect. There are a few MHC variants that have a larger effect, and there's a small number of less common variants, maybe 0.1% frequency, so 1 in 1,000 allele frequency, that have a larger effect. But most of them are actually common. And as Christophe mentioned, when we tried to do some enrichment analysis to see where these which genes would be affected by the loci and where these might be expressed, we see here, if you focus on the right side, each row is one cell. And in yellow, we have the immune cells in the data set. And then the other rows are a variety of other cells from the variety of different tissues. And you can see that only the immune cells are enriched, and the threshold significance is this black line over here. But you can see that basically virtually all different immune cells are implicated. And from NK cells, B cells, T cells. So this was really sort of expanding the previous narrative that was primarily T cells that were involved in the disease. Further, for this paper, I think one novelty was that we looked at brain. And of course, this was a big question in the field. Is MS driven by processes that start in the periphery and then get directed at the brain? Or is this some sort of process that gets triggered in the brain and then spreads to the immune system outside? And for the first time here, we looked at this with bulk data from neurons, astrocytes, and microglia. And we found that of those three cell types profiled in bulk, we only saw enrichment for microglia. And perhaps not too surprising, since these cells share a lot of genes with other myeloid cells like macrophage in the periphery. But again, this sort of points us that there may be some role for events in the CNS in the onset of the disease, which previously, again, all the other evidence was for the periphery. This led us actually, in part, was part of the motivation for us to sort of go back to the basics and try to understand this really fascinating cell type in greater detail. As we'll talk a little bit later, microglia around this time was also quite of interest because of its involvement in Alzheimer's disease. But yet, in humans, we knew relatively little about these cells because they only live in the brain. And of course, they can't be accessed until somebody is having a neurosurgical procedure or once they've passed away during autopsy. So thanks to a grant from the Chan Zuckerberg Institute, we developed an atlas of microglia by isolating fresh surgical tissue or fresh autopsy tissue and isolating all the immune cells in those samples. As you can see here, this data set, which we call the version 2 data set, and this is currently on bioRxiv, was collected from a variety of different brains of people with a variety of different conditions. You can see on the left side the different diagnoses. The idea here was not to do a disease trait analysis at this point. It was a more fundamental question of what is the variety of states that microglia can attain in different brain regions and across different diseases. So we sequenced, in the end, in this case, 74 individuals and over 215,000 individual microglia. So from this, and this is work by talented grad MD PhD student in the lab, John Tottenham, shown here, we identified this structure. And you can see here that, again, these are groups of 6 cells. Each dot is a small group of cells. You can see the different clusters that we've identified. Now, these boundaries are relatively arbitrary, of course, because you can see here that all the microglia are fairly similar to one another. So it's really sort of more of a continuity of different sort of states that are resembling each other, but can be differentiated to some extent. For simplicity here, we focused on again, these different clusters and identified 12 of them. And so with this, we went back, and this is another way to show that the different clusters. Here we have the 12 microglial clusters. The size of the circle denotes the proportion of cells of microglia assigned to that particular cluster. And then the edges are proportional to the number of cells that are ambiguously identified as belonging to either, for example, in this case, cluster 5 or 3. So there's some sort of uncertainty. And this shows that these two clusters are more similar to one another than, for example, number 3 is to number 8. But this is what we call a constellation diagram. And you can see that, again, there is a variety of different states, including an immunologically active cluster over here. For the aficionados, number 11 is one that's most enriched for the disease-associated microglia trait signature from mice, and some other features over here. So now that we have this sort of higher resolution map of microglia, we had gone beyond just getting bulk microglial RNA-seq. We were able to go back and to take another look at the MS susceptibility genes. And we can also visualize this over here on the bottom left as trajectories around different paths, potentially towards terminal fates. So if we pick this as the origin, you can see that the bright yellow are the different terminal fates that the microglia can attain. And what we can see that was interesting is that if you look at the 12 microglial clusters, 3 of them are significantly enriched for MS susceptibility genes. And they're all located in this part of the cluster. So again, clearly, so now we've gone beyond sort of implicating all microglia. It's really, part of the microglia cloud, a particular subfamily of states that are implicated in MS susceptibility. and work from a talented postdoc in the group, Verena Haag, here. So we took these signatures for different states. Again, in this case, the cluster 167, which are enriched for MS susceptibility genes, and the professional antigen presenting cell over here, number 10. And we went to an in silico analysis in the connectivity map to try to predict compounds that would create a signature, an RNA signature that mimics the signature that defines these clusters. So we had a number of about 14 different candidates. We attempted to validate them, and we validated 3 candidates, which are shown here: TORN2 and arciplastin, which both of these compounds, which have different mechanisms, polarize the cells towards this blue state over here. Again, this is the state enriched for MS susceptibility genes, while this particular one on camptothecin pushes the cells in a different direction away from this particular state here. So you can see now how we can begin to use the, going from genetic variation data towards, sort of understanding the biology of a target cell type, and, layering on compound work now so that we can identify therapeutic sort of lead compounds that can be developed further to modulate the cells and, rebalance the distribution of different microglial states. So this is how we conceptualize MS currently. So on the left side, we have genetic variation. And of course, this is the first step in the causal chain that ultimately leads to a sentinel clinical event over here. Clinically, we don't see patients until they get to this stage. Sometimes we see patients early because incidentally, they have MS-like lesions in the brain when they get an MRI for another reason. And so this is called a radiologically isolated syndrome. But at this point, the disease has already started. So ultimately, we want to go even back further and identify, of course, the events that are happening between the onset of engagement of the target organ, whether it's symptomatic or not, doesn't really matter. We want to go before the brain is involved. And so we need to understand the immune dysfunction that originates from the molecular changes that are caused by the SNPs over here. And primary prevention, of course, is the ultimate goal here, where we're trying to block progression from immune dysfunction here to target engagement. So actually working with Christoph here, we deployed our polygenic score to try to understand this other question that's bringing up here, this sort of MS prodrome. So are there a set of traits that perhaps clinically that might tell us that somebody is more likely to develop MS or maybe has early MS? And so we looked at this in the combined meta-FIWAS approach, looking at the UK Biobank and eMERGE-3 with over 500,000 participants. And reassuringly, the most significant result was MS, and you can sort of see here. Of course, this is a polygenic score based on MS susceptibility. So although these individuals were not used for that study, so it's a nice validation of our polygenic score. And the second best score is this other dominant disease. So again, most of these are going to be MS related as well. So that sort of makes sense. Also, what makes sense is that other inflammatory diseases are enriched, which again, we knew this before. So these are all nice positive controls. What wasn't actually sort of known before is sort of this poisoning of antibiotics and sulfonamide algae seems to have some shared genetic architecture with MS. This is sort of obviously some things that we need to look further into. And then some other noteworthy things that were not significant, when accounting for all the tests that we performed, was a CLL, which has been occasionally reported in context of MS, and dysmenorrhea. Anxiety was very common in MS, as is depression. And malaise and fatigue are really common symptoms for this population. But again, you can see here that these are not really sort of going to be very helpful from a sort of clinical point of view, as far as harbingers of what MS that might be coming. The other thing that we've tried to do, again, this is a nice paper by a former postdoc in the lab, Tina Verstein, who did a DNA methylation study of T cells from MS patients, mapped methylation QTLs in these T cells. And then we did a polygenic score analysis, but looking at the effect of the polygenic score on methylation of other sites, methylome-wide. And what you can see here, we found one site, which is the corticotropin-releasing hormone receptor 1 gene, that is the target of altered methylation controlled by multiple other SNPs. So 7 of the MS susceptibility SNPs seem to have a conversion effect on one CPG So one region of the genome where it was causing altered methylation. So we need to do more, but this is sort of an interesting first observation that there may be some convergence of molecular features at some point down the line. Also, we've been trying to develop new biomarkers, again, to try to characterize this difficult phase where somebody is at risk because they have a family member, for example, or they have a high genetic risk score, but they have no symptoms, their MRI is normal. So we need some measures of immune function that may be altered. And so I don't have time to get into the details here. This is a large study of 200 MS individuals and treated on glutamer acetate compared to 50 individuals. And we found some modules of co-expressed genes that were different between the two groups. So one was downregulated in MS. We confirmed this. And what's interesting is if you look over here, this is the TAN module is highly associated in MS. It's reduced. And also it's enriched for MS susceptibility genes. So here's another nice, interesting convergence that at least superficially suggests that we may have a hint about some biomarker in peripheral blood that might be a meaningful measure to monitor individuals at risk of MS. So where do we find these individuals? So 10 years ago, when we first were having some success with GWAS and MS, we were beginning to get very early polygenic scores with about 15 SNPs or so. So this is really the early days. We went out and using social media and the National MS Society, we recruited MS family members nationwide. And so we recruited over 2,700 individuals. And remarkably, over 1,400 individuals are still participating in the study, despite sort of minimal efforts on our part to keep them engaged. We're now going to do another cycle and to get sort of more individuals. But we've been following these individuals now for over 10 years. And we tested our genetic environmental risk score, where we combined not just the polygenic score, and in this case, this was 54 SNPs. Obviously, now we have a lot more. But we also included 3 non-genetic variables. In this case, sex, smoking, and infectious mononucleosis. These are all risk factors for MS. And what's shown on the right is that if you compare the groupings by the GERS, by the genetic environmental risk score, and you compare it to the middle score over here, this heptile, you can see that the individuals in the highest risk group are the most likely to develop MS themselves, and the risk is about twice that of the average MS family members. Also, of course, it's interesting that there are individuals who are going to have less risk than the average. So that can also be important information for individuals, because the baseline risk for MS is relatively low. It's about 2 to 3% for a first-degree relative. So with these tools, we can get maybe to double that risk, but we're still not that powerful and certainly not clinically relevant yet. But it was good enough to sort of select individuals who are either high or low risk in collaboration with my colleague at NIH, Danny Reich, who leads the imaging branch down there for neuroimmunology. So we flew 40 women who were high risk and 25 women who were in the low risk category down to NIH. 52 characteristics were measured, clinical measures, MRI measures, optical coherence tomography, etc. And what's shown on the left side is a plot where we're showing each dot here is one test. And you can see that while none of the tests are very significant, only one down here, which I'll talk about in a second, was significant when correcting for hypothesis testing. You can see that there's clear deviation. And so because of our small sample size, these are not significant. You can see that the two groups are different from one another. And the one feature that was significant between the two groups is the vibration sensation in the great toe, which it could indicate maybe early involvement of the spinal cord, perhaps. But again, these individuals were all asymptomatic. There just happened to be first-degree relatives. We've gone out to do more profiling of these individuals. This is a project in collaboration with my colleague and former trainee, Dr. Zhangxi Xia, who's at the University of Pittsburgh. And so we have this large project that's wrapping up to try to do a multi-omic profiling of these individuals. Fortunately, this particular figure didn't show up, but one marker that a lot of people were interested in was neurofilament light, which is a neuronal protein that gets released when neurons are damaged. But this was not very useful, at least in our group. We didn't really see individuals in our first-degree relatives that had high levels of this particular marker. But what's interesting is, for example, from the work from the microbiome profiling that Zhangxi did at Pitt, we can see that one good microbial co-abundance group is enriched for bacterial species that have been independently associated with MS as part of other studies. And it was also, this particular group was also associated with the GEMS GRS. So again, these are all sort of associations, but again, it begins to have a flavor that there's something, there may be some convergence there. So more work to be done. Finally, the question is, of course, if we want to go towards primary prevention, are these individuals actually willing to be involved in this type of research? And so last year, we queried our population and got about over 1,000 responders. And just under half of them were interested in prevention trials, which this is an important question on the clinical side. And they were interested both in vaccines and small molecule compounds. And they were also willing to participate in biomarker studies. So I think we have a population of willing participants that we can stratify to some degree. We now need to go back and improve our characterization algorithms. But again, I think there's a lot here a lot of substrate to work with. And so with MS, I think genetics has given us a lot. It's given us target genes, ultramolecular pathways, as prioritized for cellular populations to look at further. There's also evidence, from Mendelian randomization, for example, that BMI and vitamin D, two known risk factors, are also implicated through the genetic analyses. And as I mentioned, we have a committed set of high-risk individuals. But they also mentioned that genetics is not a panacea. This is not a Mendelian disease. So, the genetic risk score is informative. It's a foundation, but it's not sufficient for clinical use at this time. And but we now need to do human studies at proper scale to understand the biology better. All right, so I'm now going to shift gears to talk about Alzheimer's disease. So first, maybe I'll just sort of do a little bit of the advertisement for a project from the Alzheimer's Disease Functional Genomics Consortium. This is the first project that emerged from this consortium, about 20 different institutions, which we call the XQTL version 3 project. And Gao Wang, who is an assistant professor here at Columbia, kindly joined the analysis and is now playing a leading role in implementing this. So this builds on our previous work a few years ago, where we had integrated 3 layers of QTL information from the same brains, RNA-seq, DNA methylation, and H3K9 histone acetylation profiles, and to see whether if you have an EQTL, like an EQTL, an expression QTL, does the same SNP also control the epigenomic feature? And a lot of them do. And so this is what we refer to as an XQTL, a SNP that controls multiple levels of omic profiles. So now, again, as part of this functional genomics group, we have a large group of collaborators working together on over 12 different layers of omic data from multiple different cohorts, and we're going to do this integration. So that's just a little plug, for the group effort and for Gao, who's going to play a key role. Now, the challenge of studying the older population is, for diseases like Alzheimer's disease, of course, as we get older, a number of different diseases are becoming more prevalent with advanced states, particularly metabolic dysfunction, vascular disease, and the immune system is aging like everything else. And also, although we often think, or basic scientists often are a little reductionist and sort of think about amyloid proteopathy as Alzheimer's disease, it's not, right? It's just one of the component proteinopathies of the disease, and it's not happening in isolation. Alzheimer's related disease, which involves amyloid and tau proteinopathy, occurs in the same brain with alpha-synuclein proteinopathy, which is associated with Parkinsonian syndromes, and also with TP43 proteinopathy, which is involved in ALS, frontotemporal dementia, and other conditions. In one study of older participants, there were over 230 combinations of neuropathological features. So again, there's tremendous heterogeneity that we have to resolve if we want to really understand the disease. And so far, only 43% of aging-related cognitive decline in one cohort that we studied is explained by these known neuropathological features. So it means that half of the cognitive decline that's observed in these older individuals is unexplained. we have no molecular handle on what's going on there. So over the past 15 years, been working very closely with David Bennett, who designed and leads 2 superb cohorts out of Chicago at the Russian University. And Julie Schneider is his colleague, who's the neuropathologist for the study. This is the religious order study and the memory and aging project, ROS and MAP, or in short, ROS-MAP. So the main features here is that all these individuals are free of dementia when they are enrolled. They're followed annually by neuropsychological testing, neurological testing, imaging for many of them. And to get into the study, you have to agree to donate your brain when you die. So now, after David has been doing this since the mid-90s, and so we now have over 2,000 brains in the fridge with all the accompanying antemortem data and a structured neuropathological assessment done by Julie and her team post-mortem. So a really amazing amount of data. And actually, there's a lot more stuff here, but I don't have time to get into with post-mortem imaging and a lot of other functional characterization as well. So now, 5 years ago, for single nucleus sequencing was actually really sort of easy to do with a 10x machine. We began to work with Naomi Habib and at the Broad Institute to generate single nucleus data from these participants. And so our first data set here consists of 424 participants in these 2 studies with about 1.7 million transcriptomes. And Vilas Menon, who's also here at Columbia, and this professor, played a key role here, working together to do the analysis with us. And actually, I have a picture of him next. Masashi Fujita did really sort of the work I'm going to be describing right now, which is, the first step was just to look at the data to characterize it into the different major cell types. So those shown here, excitatory neurons, for example, are the most abundant. And you can see here that this is on the columns are individual subjects with accounts of nuclei from each type of cell. And you can see here the different cell types. So, the proportions are pretty consistent across the individuals. And on average, each individual has 3,800 nuclei. So, we did a 2-step approach because there's so many, single nuclei to analyze. So we had a cell type level deconvolution, and then within each cell type, we did lower resolution or higher resolution dissection. For example, here are microglia with a various variety of different subtypes. And you can also capture, these are relatively rare. There's only 14,000 nuclei in this data set, but we can capture all the different components of the vascular unit in the brain as well across all these individuals. So this is just, again, to highlight the microglia on the left and the astrocytes, where we now have multiple different subtypes of each. And also, this is also true for the other brain, the glial cells and neuronal cells. So Masashi, about earlier this year, began the project to map QTLs. And so the first step was to create pseudobulk data. So this is where you take each nucleus that's assigned to a particular cell subtype, and you pull them together to create a pseudobulk measurement for that particular cell type. So as a result, each donor now has 96, because we have 96 different cell subtypes. So each individual has 96 transcriptomes, each referring back to one particular cell subtype. We can then sort of map genetic effects, and we focused here on cis-QTL effects, so looking at proximal variation, genetic variation, and its effect on gene expression. What's shown here at the top is the number of genes with a E gene, so genes which are the target of an eQTL effect, and the cell type proportion for that cell. Again, not surprising, the excitatory neurons are the most numerous, sort of cells in this tissue, and therefore we have the most power to identify, E genes and QTLs, and this is shown here. The other cell types are shown here, and you have a fairly consistent sort of relationship between cell type proportion and the number of E genes. This is important because, of course, so I think we do pretty well with excitatory neurons, but we need to do better with microglia and oligodendrocyte progenitor cells here. And if you look at the cell subtypes, so this is at the cell type level. But if you go deeper and go individual cell subtypes, the relationship is even more extreme. So here you can see, again, different excitatory neuron subtypes are leading over here. and everything else is really sort of way down over here. So there's a lot more to be discovered, but we just haven't sequenced deep enough, to go to get enough nuclei for each of these rare, less common cell subtypes. What's important is also, okay, so is analyzing the level of the subtypes useful? Maybe it is, maybe it isn't. So if you look at the excitatory neurons, in green we have the set of E genes that were discovered in the bulk excitatory neurons. When you pool all these excitatory neurons together, and in gray we have the EQTLs that are found if you analyze each of the cell types, each of the cell type into subtypes separately, and then sort of amalgamate the E genes from all the different subtypes. What's important here is, you can see that a lot of them are shared, which is reassuring, and some of them are unique to the bulk excitatory neurons. Not surprising, since they're sort of pooling cells, so you're getting more transcription, better transcriptomes, and sort of more power to identify EQTLs. But what's surprising is the extent to which the cell subtypes, even though we are grossly underpowered, I think because the numbers of nuclei get really small, but we're finding a lot of EQTLs here that we didn't find in the bulk. And so I think this is really so important for a study design point of view, because it tells us that we need to go, again, much deeper in the sequencing. And this is true not just for neurons, but also for the astrocytes, oligodendrocytes, microglia. So all the cell types are showing the same pattern. We also went into our, just try to compare this to known results. You can see here comparing to a much larger study looking at EQTLs from bulk RNA-seq from the cortex, so the same brain region, but a large number, 1,000 individuals in this case instead of 400. We can see that, again, many cis-EQTLs are shared. Some are unique to the bulk, again, probably not surprising because of the much larger sample size. But even the smaller single nuke data set identifies a large number of cis-EQTLs. So again, validates the strategy that the single cell approach is going to be pretty fruitful in the brain. How relevant are the model systems that we have to work on these cis-EQTLs? So we went into a small data set of 44 iPSC lines that have been differentiated to either induced neurons or astrocytes. So these are actually lines derived from the same individuals that donated their brain. So we actually have the brain and the cell line from the same person. And you can see here that a significant number of small but significant number of E genes were found in the induced neuron and induced astrocyte data set. A little bit more in the astrocytes over here. However, when you look at the effect size, so in this case, each of the dot here is the bottom figure. Each dot is one an E gene. And you can see that many of them are concordant. So the effect size in the excitatory neurons is the same from the brain, is the same as the effect size in the direction in the induced neurons. But you can see here there's actually a substantial number of E genes that are having opposite effects. And one of them, which is important, is MAP-tau, which is actually the gene that encodes the tau protein. So obviously of major interest in Alzheimer's disease. And this is shown over here. Here's the data from the astrocytes. You can see the particular SNP over here. So it's the same SNP for all data. Because astrocytes, excitatory neurons, same effect, same direction, but opposite direction for both in the induced system. So it just shows that these effects may exist in the model system, but we have to be very careful and to look into the direction of effect as well. We also have done sort of the standard co-localization analyses. So this is with the Alzheimer genes. And what's shown here, this is the posterior probability that the SNP that is associated with susceptibility is also the SNP driving the expression in QTL. And so the dark color denotes a high probability that the two are co-localized or concordant. So it's not a coincidence. And at the top here, we have, again, we did the different cell types. And you can see that the cell type that has the most sort of co-localization are the microglia. Not surprising because microglia enrichment in Alzheimer's disease genes has been noted many times before, but this is really, I think, the first time to show this co-localization effect. But importantly, we also see this in excitatory neurons, in oligodendrocytes, as well as the other cell types. So in Alzheimer's, again, microglia definitely play an outsized role, but the other cell types are implicated as well. And we can use, these data to create genetic instruments to infer expression, to do a TWAS. This is a microglial TWAS in a large data set from the that was published, and we can see a number of different loci that emerge, some of the known factors and some that are actually not picked out by the GWAS. So, okay, this is just to illustrate the different things that we were able to do with our EQTL mapping. Going back, of course, we can also look at how the frequency of each of these different cell subtypes, and we have 92 different cell subtypes, we can see whether they relate to the different traits that we have in these individuals, including the amount of amyloid in the brain, the amount of tangles. So these are the two cardinal features that define the disease, amyloid and tau pathology, and also the rate of cognitive decline. And you can see here that overwhelmingly, microglia seem to be the cell types that are changing the most in the disease, but astrocytes also are close behind, although there seems to be a difference here. Again, this one, for example, not being associated with amyloid, but just tau and cognitive decline, whereas microglia 12 may have more role in the pathology, but it doesn't seem to have much of or not associated with cognitive decline, while this particular microglial subtype is associated with all three. Since we didn't have a properly powered replication data set, what we did was to take bulk RNA-seq data from other brains in the same collection. So we had another set of brains over here, 684, total of 1,092 brains in this collection. So with this, we were able to infer proportions of each of the cell subtypes in the bulk RNA-seq data, and then to repeat the analysis in this replication data set, where again, we're not measuring single nucleus data directly, but we're inferring the proportion of subtypes using the CellMod algorithm that Vilas Menon developed for this purpose. And this works quite nicely, actually, and you can see here with the meta-analysis where we combine the single nuc data with the inferred bulk data, the microglial 13 and astrocyte 12. I mean, the top results remain, and we now have many other results that are sort of perking up also with the increased sample size. Maybe more convincingly here, what I show is the effect size for the single nuc data sets on the data set on the x-axis and the inferred proportions from the bulk RNA-seq on the y-axis. And so you can see the results are fairly consistent. There are some differences. So you can hear clearly for the microglia, which are less common cell types, so probably less well inferred in the bulk data, they seem to be underestimated, in terms of the bulk, compared to their true effect in single nuc data. But the astrocytes actually are doing pretty well. So the effect size is quite similar in the two data sets. This is for amyloid in relatively few associations, more associations with tau pathology, so more cells are changing in association with tau, and cognitive decline as well over here. Now, what else can we do? Because of course, associations are nice, but Alzheimer's disease is a disease that evolves over decades, that has multiple different stages. In general, we're not going into the details, but we think that it starts with amyloid proteinopathy that accumulates, eventually tau proteinopathy accumulates, and then later cognitive decline occurs. And so we can use structural equation modeling to try to understand where these cell types that we've implicated as being associated with a particular trait may be having a role. For example, microglial 13 over here was associated, if you remember, with amyloid, with tau, and cognitive decline. So it could really be almost anywhere, in this sequence of events. But our modeling suggests that it's somewhere downstream of amyloid. It mediates part of the effect of amyloid on tau proteinopathy, but not the whole thing. And then tau itself sort of goes on to affect the astrocyte 10, the effect size, which then mediates the effect of tau on cognitive decline. So now again, we can begin to position the different cell types. And so this is important to address targets, of course, for different purposes. If you want to intervene in tau proteinopathy, microglial 13 is probably a good target. Astrocyte 10, probably not so much. But astrocyte 10 might be very relevant if you have something that prevents cognitive decline caused by tau pathology. We can also use these data to project individuals in two-dimensional space. So this is using the fate algorithm. So here we're using the frequencies to project individual subjects. So each dot is one person or one participant in the study, and in the sort of two-dimensional space. And so this is what the projection looks like. So it's interesting. You can really see obviously two arms to this distribution. And you can see here different subtypes, for example, microglial 13 that I highlight as being very associated with pathology seems to be enriched in this part of the distribution, as is, oligodendrocyte 13, for example, and astrocyte 10 particularly seems to be very enriched. So this is one way to visualize, these different trajectories. And astrocyte 5, for example, is very enriched in this particular branch. Now we can also relate this to the different traits. So if you're looking at amyloid, there seems to be a lot of amyloid accumulation in individuals who are in this part of the distribution, and this what we call the southern arm or the southern distribution versus the west over here. If you look at tau pathology, it seems to be more down here, a little bit in the middle, but more to the south. And particularly cognitive decline is also stronger down to this branch. So what we can do is to really create what seem to be two different trajectories. If you take each individual as one point along a trajectory of aging, we can see two paths. Perhaps one path going south or down that seems to be enriched for pathology, and one going west, which is there's some enrichment for pathology, but not as much, and not as much cognitive decline. And this is also, so you can use these to create pseudo trajectories. So if you're looking at this particular trajectory at the traits, you can see that as people go from the source to the distal point, amyloid accumulates, but not, that's not dramatic at the end. Tau really is monotonic and sort of increases all along this trajectory. And cognitive decline sort of follows this pattern here, increasing somewhere in the middle, and maybe tailing off a little bit at the end. We also can see whether some of these cell types are correlated with one another. So here we're just showing a correlation matrix based on the frequency of the different cell subtypes. So we're able to group different subtypes, for example, some astrocytes with microglia and oligodendrocytes, so to communities of cells that appear to be correlated in frequency in these brains. Because obviously the tissue is not changing at the single cell level with individual cells, but rather the community cells that are interacting with one another. And so this is what we see if we remap, some of these associations. We're really seeing sort of two main communities of the cells that are really the reciprocal of each other and shifting back and forth, between these being enriched in disease or being enriched in the people who are more resistant. So where we are right now is, again, we've continued, just like with MS, we have this trajectory starting with susceptibility variants, leading to clinical syndrome, here Alzheimer's disease. And as I've shared with you, we're beginning to have elements along the way of what seems to be connecting these different pieces of the puzzle, including amyloid pathology, tau pathology. And for microglia, for example, it seems that one component is very important for tau pathology, but another one may be important for amyloids, the same cell type being involved in different processes in parallel. So again, what we've learned with the single nucleus RNA-seq data, we've learned a lot. We're just starting to scratch the surface with this. We prioritize the microglial 13 astrocyte 10. But again, lots more remains to be done. The gene mapping, as I showed you, is really a first draft. We need to go much larger in terms of the sample size, but also, even more importantly, deeper sequencing to get more nuclei per individual to really get at the subtype level effects. And again, I mentioned the iPSC models, which seem to preserve some of the effects, but the progressionality may not be consistent, so we have to be careful. Now, I'll tell you the last part. How much time do I have?
10 minutes.
Okay, that's fine. So the last vignette is about immunosenescence. So again, some of the connection here, of course, is the immune system and how it's related to cognitive aging. Immune aging is a feature as we get older, and what some of these changes that happens are shown here. The thymus involutes early on in life, and over time, at midlife, things are relatively stable, but then begin to decline after age about 50. And as we get older, as we saw with COVID, their susceptibility to infection increases, incidence of certain chronic diseases, cancers, and certain inflammatory diseases increases. And more we're beginning to appreciate the role of inflammation not as a reactive element, but really as a causal element in some neurodegenerative diseases. Again, different pathways, and this is still not well elucidated, may be involved in the different diseases. So this is work of Hanan Tool in the group. And this is working with colleagues here in neuropsychology, which had a terrific study called the RAND study, where they were looking at cognitive reserve across the entire age spectrum. So they had already sampled individuals from 25 to 88 years old. We collected blood from them, and we can now relate, the immune profiles that we've generated to neurological examinations, cognitive examinations, and MRI. Also, very importantly, we used some subjects from the YCAP offspring cohort to enrich our sampling of individuals who were of diverse origin, including African Americans and people who with Hispanics. And these are all self-declared ancestry. So I'll talk about the cytometric profiling that we've done so far. We've also done other forms of data that I won't have time to talk to today that we're still in the process of analyzing. But we basically have a multimodal assessment of peripheral immune aging that's coming. So on the right side here, we have some classical cytometry data. This is where we're projecting the data into 2 dimensions, in this case, C45RA and C287, 2 markers on the cell surface of the different immune cells. And here are the naive CD8 cells. Each dot here is 1 cell. You can see the distribution of this particular group of cells seems to be quite high in both markers. So this is a young individual. You can see there's relatively few cells over here. These are the type of memory cell called TEMRA cells. On the other hand, if you look at a 69-year-old participant, you can see a clear visual shift with a lot more cells being present here proportionally than up here. So this is sort of the classical way to segment cytometry data by doing 2-dimensional gating sequentially by hand. Not the most efficient system, but this is sort of the traditional one that emerged in the 1970s. But with this, we're able to reproduce known associations with, again, naive CD8 T cells decreasing over age, so age on the x-axis here. These TEMRA cells are increasing with advancing age, as shown over here. Importantly, we have the 3 groups. We have non-Hispanic whites in blue, African Americans in yellow, and the Hispanics in gray. And you can see that for these particular cell types, the 3 groups are largely parallel. So there doesn't seem to be any population-specific effects for these particular cell types. We did a meta-analysis approach. So we did a separate analysis for each of the 3 groups. Again, using simply the self-reported description of the individual, and then we meta-analyzed across the 3 different population groups, and we find 14 different markers that are populations of cells, rather, that are significantly associated with aging when controlling for sex and technical factors. But when we did find some new findings also. So maybe I'll just mention it here. I won't have time to get into it, but what's interesting in this particular analysis, some of these populations have an interaction by a population, meaning that some population, particularly the Hispanic population, behaves quite differently from the other 2 populations in terms of the trajectory. They have a more marked change with aging than the other groups. This is using the self-reported ancestry. Now, separately, we also use the data. So this is low-dimensional data, but 14 different colors, so 14 dimensions for each cell. And we had 2 panels, so one of close to 4 million cells, one with 3.6 million cells. And we use the same tools that we use or similar tools that we use for single cell analysis to analyze this data. And so we're able to cluster them because these markers are highly informative. And we're able to identify 39 clusters here, and for this panel, another 32 clusters of different cell types, which capture on the left again, capturing the major different cell types in the peripheral blood, and here focusing on the T cells on the right. So this is work that Tanya Luques, a PhD student that's co-mentored by myself and Vidas Menon, has been doing. Really terrific work to use a phenograph to segment these data in an automated fashion. So again, you can see here the different markers in rows, different cell types that he identified in columns. And then there's a lot of information here. I'll just skip to the bottom, which is where the money is. So this is the effect, the associations of each population with age. So you can see here, these 3 populations are significant when accounting for multiple hypothesis testing. these are different NK cell populations. There are also some CD8 populations that are significant, and that's about it. Tyne also did a separate analysis, which unfortunately I don't have time to get into, which is really looking not just at the level of the individual subject, but looking at the individual cell and its probability of existing in a certain part of the multidimensional space at a given time. And that analysis also gives us some additional findings, some of which are consistent with the association with age, with using traditional linear regression, and some, again, that are different that emerge here. And interestingly, shown over here, in this case, we had done genome-wide association, genome-wide genotyping in each individual, and we were able to identify the principal components that map each individual along the matrix lines of human populations, with PC1 being the main European African cline, and then the APC2 really pulling the Amerindian component out of the Hispanics. And we see here that actually largely PC1, so this cline between Europe and Africa, there's really only one population, a type of B cell that seems to be affected by your level of ancestry along this gradient. But the other populations are not really affected genetically. This is different from what we saw in the self-reported ancestry, where the Hispanics looked quite different for several cell types. And so we have now sort of a disjunction between the genetic analysis and the self-reported analysis, suggesting that some of the findings may be due actually to other factors that are non-genetic, but correlated with a particular self-reported ancestry. Again, more work needs to be done, obviously. Finally, Natasha, here another postdoc in the lab, used the FATE algorithm, which we saw earlier with single nuke data, to plot individual subjects in two-dimensional space based on these data. This is shown over here. Again, here we have the non-Hispanic whites, non-Hispanic blacks, and the Hispanics over here. Everybody's sort of distributed across the distribution, and you can see some, this is colored by the age at which the sample was collected. You can see maybe some gradient here about with age. There's clearly another factor that's driving some individuals in terms of their immune profile in another direction. So again, more work needs to be done here to understand these different trajectories. We can also look at the correlation structure and how and which factors are the most important in trying to predict age. And actually, with these features, even though we only use one panel in this case of cytometric data, we can explain a little bit more than about 60% of the variance in chronological age using these data. And so with this, we developed an immunological age estimate, which is shown over here. Here's a chronological age on the x-axis, immunological age on the y-axis. And you can see here each dot is one individual. It's a little bit noisy, but you can see that there's, again, there's a correlation, obviously, but there's quite a bit of noise. So people are going away from the line. And this delta is what we refer to as the immunological age burden. If it's greater than 0, this is a suggestion of accelerated aging potentially of your immune system. And if it's less than 0, so perhaps slower aging. Interestingly, this score was associated with brain volume in a subset of individuals where we had MRI volumetric data available. More needs to be done here. And what's shown here is that the immunological age, which we initially designed based on the non-Hispanic whites, because that's the largest group, and we sort of tried to do that first on the largest data set. If we apply it to the Hispanics, which again are quite different, then you can obviously see that the Hispanics don't have the proper behavior of this immunological age calculation if we're using the one developed for the non-Hispanic whites. So highlight, in fact, I think pretty obvious, but that we need to personalize these immunological age and really take into account the variability that's occurring in the different populations. So with this, I'll close and so just sort of summarize this part of the talk. So we have this multimodal PBMC profiling that can be done at large scale. And we have, again, single cell data that's coming with some also some functional data from T cell proliferation. So more to come there. As I mentioned, we have this interesting disjunction between the self-reported ancestry and the genetic ancestry that needs to be investigated further, but it suggests that there probably are so non-genetic environmental factors that are causing some differences. And I think what's actually quite exciting for me here is that this type of cytometry is clinically deployable. These are tools that already exist in the clinical lab, doing this a panel such as this would be easy to do and could be potentially useful, for a variety of applications, whether it's to stratify risk for infections, for example, as with COVID, or with for cancer immunotherapy. I'll close and then I'll try to recognize. I already recognized most of the people as I went along. Again, Masashi did a terrific job on the single nucleus data. Verena and John, who have worked on the microglial story, and Natasha and Hanan and I don't have time because he's technically part of Vilas' lab, but obviously co-mentored as well. Did a terrific job on the third story. I'll stop there. Thank you very much.
Thank you very much. This was an amazing talk and amazing data sets that you generated for all three different projects that you described. So the session is open to questions. Please raise your hand if you're in the if you're online.
Question over there?
Yeah, thank you so much. That was such an informative presentation. I have a question about the Alzheimer's disease part of it. You mentioned cognitive decline. I was wondering if that was just episodic memory or working memory.
Yeah, so we use it's an excellent question. We use a construct that's a summary. We a global cognitive score that's derived from 19 different neuropsychological measures, which are overrepresented or overweighted by episodic and semantic memory tests. In general, what we've seen is if you so within that is like there are 4 tests for episodic memory, for example. If you look at the episodic memory separately, usually it tracks with this global cognitive score. So we in general take the global cognitive score, which is then repeated annually, and from there we get this linear, the slope for linear regression, which is actually not ideal because the true trajectory is not linear, but it accelerates 3 years before death. But again, it's a reasonable approximation.
Perhaps as people are thinking about their questions, I'll ask one too. Could you comment on the effects of polygenic risk scores in Alzheimer's disease and multiple sclerosis, and kind of what are the predictive properties of these scores? What is the SNP-based heritability for these phenotypes?
So again, for MS, we showed you, I think that there's some utility to stratify risk for family members. And then again, this is for susceptibility. So when we try to actually relate a polygenic score based on the MS GWAS, again, this is a susceptibility GWAS. If we look at the disability after MS starts, there's no correlation. So it's actually an interesting observation that whatever genetic architecture is relevant to the onset of the disease doesn't seem to be relevant once the disease starts. So that's for MS. For Alzheimer's, there's been a number of different studies. There are relationships between polygenic score and brain atrophy, like hippocampal atrophy, for example, probably with cognitive function as well. I don't recall specifically, but most likely there is that as well. What's interesting now is the number of variants has increased. Actually, one of my colleagues, Steve Yang at Brigham, has developed a microglial specific polygenic score, so trying to be more coherent about the risk due to that particular cell type, and shows that there's actual relationship between that score and some of the microglial features that may be relevant to the disease. So I think we're going to probably see some more of that as we understand better which variants work together.
Thank you. Okay, and other questions online? I might just actually make a comment about the GWAS for multiple sclerosis polygenic risk score. I think one of the reasons why the fatigue and malaise did not get much in terms of statistical significance is the fact that physicians typically don't code symptoms very well using ICD system. So perhaps that's why these symptoms are not coming up.
That's a good point. I agree. I think these are going to be more noisy traits.
A question. So in your summary about immunosenescence, there was a very strong statement that caught my attention about Hispanic participants that display more pronounced change with aging. So how do you correct for things like lifestyle, like, maybe drinking or smoking, or it's like for us radiologists, a lot of times that's the problem we have with those imaging. It's like, how can you say that it's related to normal aging versus like some other factors like small vessel ischemia or smoking or something like that. So
it's a great point, and I think this is what we're trying to do now, because again, this cohort was recruited for a particular purpose to look at cognitive reserve. So they have a variety of traits, but not necessarily all the ones that you would want. But we are going to take a look at things like diabetes and smoking and some of the obvious things, but there's a lot more variability, of course. So we are going to try to look at other factors like that you can get, for example, from the zip code, right? Some environmental data, or also the disparity index. So that's been calculated to try to relate, to try to get a hint as to what element of the environment or the non-genetic component may be at play here. But yeah, it's very important and fascinating, but very difficult, as you know, to study. So we don't really measure a lot of these variables very well, or we don't collect them.
Are these patients recruited here at Columbia, or are they?
Yes, they're mostly from the population around the hospital. I think again, this is a collection from a different from a colleague that recruits, but they recruit by mailing out to the surrounding people in the surrounding zip codes. So it's a relatively random sample of the population.
I'm really impressed by the degree of correlation of immunologic age with the chronological age, with R of 0.8, and over 50% of variants explained. That's pretty impressive.
Yeah, although I agree, but this is the same data set, right?
Yeah.
So I think we're now actually got funded to do a much larger study. So this is 250 people. So we're expanding this to 1,000 people with 3 time points over 4 years. This is all cross-sectional at this point, but we'll have longitudinal data and we'll have more individuals, and that's leveraging a different cohort, the YCAP cohort that Dr. Mayu developed years ago. And by chance, that population actually has an overrepresentation of Hispanics. So I think we'll be able to get maybe at some of these questions they have. It's about 60% Hispanic, 20% African American, 20% non-Hispanic. So yeah, I think we're in the process of collecting the biological samples at this point.
Do you have any ideas of what's driving these immunologic changes? Is it cumulative pathogen exposures? What do you think is happening?
It's a great question. Yeah, so it's not clear. I mean, I think part of it is that until about age 50, you don't really see that much difference. It's really what's interesting. Maybe I don't have to think about it anymore. But the variance in the frequency of some of these population really explodes in later life. So it's quite tight until about age 50 or 60. And after that, there's a lot more variability between people. Part of this may reflect the features of exhaustion. And I think so again, not showing here, we also profiled some people with MS to see what they look like in terms of immunological lesions. Of course, they have a chronic inflammatory disease. Presumably, they're going to look older because there's probably some elements of exhaustion into their immune system. But this is where, obviously, now that we have the booster, like the initial version of our tool, we want to deploy it in a number of different populations to see how it behaves in the disease population. For example, one example I mentioned is the people going for therapy for cancer. Is this a relevant trait or not? So maybe that might help to prevent adverse events that are basically due to lack of regulations and the responses.
I was also thinking to what degree COVID infections or repeated COVID infections contribute to immunologic aging.
Yeah, well, that's true too. Okay, this is all actually samples collected before COVID. So yes, I think the next set is going to be quite good.
Okay, any other questions?
I have another question. Sorry. I'm not sure if I missed this, but you had some really interesting imaging data for the MS part of the presentation. I was wondering if you had found the information that you found with the tauopathies and the level of the amyloid and some biomarkers. You found any interesting correlations to the hippocampal volumes or prefrontal cortex volumes?
Yeah, so we haven't done too much with the volumetric measures. Yeah, to be honest, we have a good problem having too much data. But we actually, there are some interesting, we have a different study where we looked at monocyte RNA-seq from these older individuals. And we do have actually several modules of co-expressed genes there that correlate with hippocampal atrophy. So suggesting that, again, it's modest. I would say even with 200 individuals, we barely have significance, but it's there. So there are things going on in the periphery that are related to what's going on inside the brain. Again, not surprising since the peripheral immune system is communicating constantly with the central nervous system, immune system. So the two are yoked, obviously, to various degrees. But if you're interested in the data, is freely available. So all the data is downloadable from the Neurologic portal by NIA.
Okay, thank you very much. I don't see any additional questions. Thank you for a wonderful talk.